Professor Larry Chamley. But I think we'd give people a few more minutes to join because I think everyone's probably glued to the conference proceedings. Sorry, to Jacinda Ardern talking about what level 2 might actually mean for us if we do go into level 2 next week. So let's just give a couple of minutes for people to swap between what they're doing. But today, Dr. Debina Sarkar, sorry, have I pronounced that correctly?
It's all good.
is going to be presenting a research paper to us. So let's just give people a couple more minutes and we can start.
Cool, thanks, Sandy.
Dr. Debina Sarkar, she's a postdoctoral research fellow at the Department of Biochemistry in the University of Otago, and her area of interest is cancer genetics, looking at long non-coding RNA. So let me just hand over to Dr. Sarkar, and she can talk to us about her research journal, sorry, her paper that she's presenting.
Cool. All right. Hi. Yeah, I kind of came across this paper and I was like, oh, that looks like a cool paper. And then I was like, holy shit, that's a lot of figures and experiments that they did. So yeah, I'll just see how I get on with it. So as Sandy said, I am in Sarah Diermeyer's lab down in Otago at the Department of Biochemistry. And we are not experts in EV per se, like we want to get into the field, so pretty much have no, I wouldn't say no awareness. I know what the challenges are of going into this field, but trying to get our feet wet and things like that. And yeah, so I thought this paper was very cool in the sense that this is the first mechanism that I found that wherein EVs are actually acting as cell signaling molecules, which was quite cool. And to top out the fact that it actually involved the lncRNA or long non-coding RNAs that are we work on them every day. That's what our group specializes in, and that's what my background is in. So it really kind of piqued my interest. So with that, I'll just start off. Trying to share screen. Can you guys see my screen, hopefully?
No.
You can't?
No.
I don't know what I did. Hang on. You see it now?
Yep.
Hopefully. Good? Good? Awesome. Okay, so the title of the paper is very long. So it's an extracellular vesicle packaged HEV1 alpha stabilizing lncRNA from tumor-associated macrophages, which regulates aerobic glycolysis of breast cancer cells. So a little bit of a background. So I haven't included anything about lncRNAs, but then for those of you who are not really, I'm sure everyone knows about it, but then just to give a brief background. So long non-coding RNAs are RNAs that do not code for any proteins, and what makes them special is their tissue-specific expression, and they are normally more than 200 nucleotides in length. So with background for this specific paper, basically there's diverse mechanisms that reprogram glucose metabolism of cancer cells in different areas of the tumor. And this glucose metabolism is kind of regulated by this hypoxia inducible factor that's a transcription factor, which control the protein levels of which are known to enhance aerobic glycolysis in tumor cells. Limiting factors of HIF1 alpha include PHD2, which causes hydroxylation and proteasomal degradation of HIF1 alpha. And this depends on the tumor microenvironment, which is again regulated by factors like hypoxia, reactive oxygen species, nitric oxide, and certain other metabolites. The other players that come into regulation of aerobic glycolysis include the tumor-associated macrophages, which are known to promote malignancy and regulate tumor cell glycolysis. So the main questions that were addressed in this paper was whether TAMs were involved in regulating aerobic glycolysis of breast tumor areas, specifically ones that are infiltrated with macrophages. And this group previously studied TAMs, wherein they found that TAMs release EVs to transmit microRNAs into adjacent tumor cells to regulate their biology. And the other main question that they addressed was whether whether link RNAs packaged in EVs from TAMs regulate aerobic glycolysis of cancer cells, and what are the underlying mechanisms that regulate or that are responsible for that. So in order to see if TAMs actually enhance glycolysis and reduce apoptosis of breast cancer cells, what they did was they used a panel of breast samples or patient breast cancer samples, and they stained them for CD68 and CD68 in addition to staining them for GLUT3, GLUT1, and HK2. So GLUT3, GLUT1, and HK2 are markers for glycolysis or enhanced glycolysis. So as you can see from this panel here, so the cases wherein there's low CD68, so CD68 is a marker for TAM, and TAM. So high CD68 corresponds with high expression of GLUT3, GLUT1, and HK2 compared to CD68 low samples wherein you don't see any increased levels in the glycolytic markers. In parallel with this, they wanted to see the level of apoptosis in tumor samples which were high in GLUT, or which expressed high levels of GLUT3. And as you can see from the figure here, so they did a tunnel assay, which is essentially assesses the amount of DNA fragmentation. And as you can see from here, high levels of tunnel wherein DNA fragmentation is quite high, resembling apoptosis, is quite high in samples wherein GLUT3 was quite low compared to high expression of GLUT3 when you don't see any apoptosis that much. So with this, they proceeded forward to study the association between TAMs and breast cancer cells. And the approach that they took was they developed or they isolated something called as the pre-TAMs. So pre-TAMs are basically primary TAMs, which were isolated for human breast cancer tissues. And post-TAMs, basically what they did was they treated the human monocyte-derived macrophages with conditioned medium of MDA MB231 cells. They call this polarized TAMs or pol-TAMs. And then following this, what they did was they co-cultured the MDMB231 cells with either pre or pol-TAMs in a transwell system so that they are not in direct contact with one another. Following this, what they saw was in the MDMB231 cells that were co-cultured with pre-TAMs, they saw increased levels of glucose consumption compared to the 231 cells that were co-cultured with just the MDMs or the ones that were left cultured by themselves. The same was observed for lactate production. So 231 cells, which were co-cultured with pre-TAMs, showed high levels of lactate production compared to the ones that were co-cultured with MDMs or were cultured by themselves. Same was seen for extracellular acidification rates, or ECAR rate, was found high in the 231 cells co-cultured with pre-TAMs. In addition to this, they also did some Western blots for specific glycolysis markers for GLUT3, HK2, LDHA in the 231 cells, and found that the cells were co-cultured with pre-TAMs, the levels of these glycolysis markers were quite high. Following this, following on from the apoptotic story, wherein GLUT3 levels, high levels of GLUT3 corresponded to low levels of apoptosis, what they did was they treated the 231 cells with docetaxel, and they found that the apoptotic levels dropped significantly in 231 cells co-cultured with Pol-TAMs compared to MDMs or the cells that were cultured by themselves. So that kind of gives a good correlation between TAMs and how there may be a relationship between the breast cancer cells and the TAMs, or the infiltrating macrophages. To carry the story further, they wanted to see if HIF1 alpha played a role in this interaction between TAMs and breast cancer cells, and if TAMs actually influenced HIF1 alpha to enhance aerobic glycolysis and inhibit apoptosis. So what they did was they used CHX, which is a transcription inhibitor, and then did a Western blot or immunoblotted for HIF1 alpha in 231 cells. So, and they used untreated versus 231 cells that were cultured with TAM culture or conditioned media, and found that HIF1 alpha was present, the half-life of HIF1 alpha was quite high in case of breast cancer cells that were conditioned or that were cultured with the TAM conditioned media. So that gave a clue that HIF1 alpha might be playing a role in enhancing aerobic glycolysis. Following this, what they did was they knocked down HIF1 alpha using siRNA and found that the ECAR rates decreased in 231 cells that were co-cultured with TAMs compared to the negative controls. And the same story followed on for in case of glucose consumption and lactate production. So 231 cells wherein HIF1 alpha was knocked down using siRNA and co-cultured with TAMs, glucose consumption reduced, and same was the story for lactate production. And they also did immunoblots or Western blots for glycolysis specific markers and found that levels of GLUT3, HK2, and PKM2 were reduced where HIF1 alpha was knocked down. And the same story for apoptosis, wherein 231 cells treated with docetaxel, but then in 231 cells that were in which HIF1 alpha was knocked down, they showed increased apoptosis. So that kind of followed along the story. So the story is still holding up that TAMs influence aerobic glycolysis maybe through HIF1 alpha. Next, they wanted to see how TAMs are actually enhancing aerobic glycolysis. So they wanted to see what factors or what mediators were playing a role in actually causing this enhancement. So what they did was they fractionated the components of the Pol-TAM culture media. And then this is what they did. So they did ultracentrifugation of the TAM culture media. And then again, exposed the 231 cells, wherein they were treated with culture media from the Pol-TAMs, from the supernatant, following the ultracentrifugation or the pellet. I'm sure people will have concerns about, I'm not an expert, again, would be good to like see how people take the ultracentrifugate, like how EVs were isolated. I am aware that Cherie is not a big fan of ExoQuick, so it would be good to hear what they have to say about this. But anyways, they ultracentrifuge the conditioned media and obviously expose the 231s to culture media, supernatant, or the pellet. And then they found that when the 231 cells were exposed to the pellet or the conditioned media is when the glucose consumption and lactate production bumped up. What they then did was they used the pellet, resuspended it, and then EVs isolated from the conditioned media. And they visualized it using TEM, electron microscopy, which is here. And then they assessed the diameter or the particle diameter or EVs using nanosite. And they immunoblotted for certain EV-specific markers like Alex CD63, CD81 in the POLTAM or EVs and supernatant. So they were present in EVs and the cells, but not in the supernatant. Following this, what they did was, in order to see that these EVs were actually internalized by the breast cancer cells, they labeled the EVs and then used RAB7 late endosome markers to see the internalization of the EVs. As you can see here at 90 seconds, there's co-localization of RAB27 and late endosomes and EVs, indicating that the EVs were internalized in the breast cancer cells. Following this, they wanted to see if it was an EV specific thing. So what they did was they treated the conditioned media with anti-CD81, which neutralizes the secretion of, it depletes EVs in the CM of TAMs. So they cultured the 231s with these different treated TAMS conditioned media, wherein they used untreated IgG and anti-CD81, which depletes the EVs, and found that when 231s were cultured with TAM EVs, where the ECR activity went up. The same was the case for glucose consumption and lactate production. So they then wanted to see what was in these EVs that was causing this change or the increase in glycolysis. They followed it up with a lncRNA profile of different samples. So they compared, so they did a microarray. And for lncRNAs, wherein they compared MDMs versus TAMs, the macrophages, so polarized versus the MDMs. And then I haven't shown you the heat map for the lncRNA, but then just put in selected figure. I'll get to it in a minute. And then, and they compared lncRNAs from MDM EVs versus lncRNAs in the TAM EVs, and finally compared the differential expression of lncRNAs between the untreated MDM B231 cells to 231 cells that were treated with TAMs. And they validated the microRNA data using QRT-PCR, wherein they confirmed that 5 out of 7 lncRNAs were increased by more than 5-fold in EVs from POLTAMs and by more than 3-fold in the 231 cells co-cultured with the POLTAMs, as you can see from here. And then this is the experiment that they did, wherein they were sure that the lncRNAs were coming from EV and not anywhere else. So what they did was they co-cultured, so inhibition of EV secretion abolished. So they treated the TAMs. So they had SH-RAB27, shRNA against RAB27 that will deplete or inhibit the production of EVs. So that would mean that the 231 cells wouldn't get the EVs. And as you can see from here, the 231 cells wherein they were left untreated or treated with TAMs, you see a high expression of HESLA compared to the 231 cells wherein they were co-cultured with TAMs wherein RAP27 was inhibited. They then wanted to see if HESLA was responsible for increase in the glycolytic activity. So they did several experiments for that. So initially what they did was they silenced HESLA in the TAMs, and then as is evident from here, they found that the ECR activity decreased in the 231 cells, wherein where HESLA was knocked down. And the same effect was replicated in this experiment, wherein they inhibited RAP27, which inhibited the secretion of EVs. And you can see here that the ECAR activity decreased in the 231 cells when they were co-cultured with TAMs wherein RAP27 was knocked down. The next set of experiments included targeting of HESLA in the breast tumor cells, breast cancer cells themselves, wherein you can see that TAMs the ECR activity was reduced in the 231 cells. And then the same effect was, sorry, not the same effect, but then a rescue assay was also done wherein ectopic expression of HESLA brought up the expression, brought up the ECR activity. Following on from the same story, they saw reduced or increased apoptosis when HESLA was knocked down in the TAMs when co-cultured with the 231 cells here. And then the rate of anoikis was also more when you knock down HESLA in the TAMs in the 231 cells when they were co-cultured with 231 cells. And then they also saw that they partially reversed the effect on migration and invasion. So the migration rate reduced when HESLA was knocked down, and the same for invasion as well in the 231 cells. They also did an absolute QRT-PCR to figure out how many molecules of HESLA were present in EVs from MDM versus TAMs, and how many HESLA molecules were present in the 231 cells in here when they were co-cultured with TAMs versus the ones that were left untreated. This I found quite interesting because in itself, the number of molecules in TAMs is around two. But then here, where in 231 cells are co-cultured with TAM EVs, they seem to have gone up to 270, which was quite interesting to me. And then this essay was again to see if HESLA is actually being taken up by the 231 cells. So similar to the experiment that they did before, they labeled the HESLA with biotin marker, and then label the RAP27 late endosomes, and after 300 seconds you can see the co-localization. So that indicates in the 231 cells, that indicates the internalization of the EVs from TAMs into the 231 cells. They then wanted to investigate what was driving up or what was regulating the expression of HESLA in TAMs. So they started with some, so they used Jasper to figure out if there was any specific motive in HESLA that was that kind of that was enhancing the transcription factors to bind onto it. So they found a binding site for in the HESLA promoter for PU1, which is again a transcription factor and a key regulator for myeloid and B lymphoid cell development as the promoter of HESLA. And they validated that using CHIP assay, wherein and followed by PCR using an anti-PU1 antibody, which confirmed that the binding of PU1 was more at the HESLA promoter, and they used CD11B promoter as a positive control. And this was done in the THP1 cells. Following this, they wanted to see the binding specificity of PU1 to the binding site or the motif. So what they did was they introduced mutations in the binding site and found that the mutation actually affected the binding of PU1 and therefore decrease in the luciferase activity of the HESLA promoter. Just kind of validating this further, they knocked down the expression of PU1 in MDMs and TAMs and found that HESLA expression decreased on knockdown of PU1 in both MDMs and TAMs. So all of this suggested that PU1 was responsible for a selective HESLA transcription in macrophages. They further wanted to see what was the stimulant or the trigger that actually drove up HESLA expression, because they found HESLA expression higher specifically in pol TAMs compared to MDMs. So they're higher in conditioned TAMs compared to MDMs. And one of the things that they wanted to investigate was if lactate secretion by the breast cancer cells was actually driving the expression of HESLA expression. That is exactly what they did in this experiment. So they used 231 conditioned media, that is MDM231, MDMB231 conditioned media, which they treated with anti-GM CSF, so which is a granulocyte macrophage colony stimulating factor. So that was one of the factors that they thought may be affecting HESLA expression. Or is it lactate that is actually driving up? So they used the inhibitor against lactate secretion, which is oxamate, and DCA, which are inhibitors of lactate secretion. In addition to this, they also did some control experiments for 231 conditioned media to see if EVs from 231 was actually influencing HESLA expression. So what they did was they again used anti-CD81 to inhibit the secretion of EVs to deplete the EVs from the 231 conditioned media. So here you can see that when you treat 231-CM with anti-GM CSF, there is no difference, the HESLA expression is not affected, but when you inhibit the secretion of lactate, absence of lactate definitely affected the expression of HESLA. So this indicates that lactate release from the breast cancer cells stimulated HESLA production in pol TAMs. Next, what they did was they knocked down HESLA in pol TAMs to see if that affected ECR activity rate in pol TAMs themselves. And they found that when they used 231 culture media, but they knocked down HESLA in pol TAMs, it affected the ECR activity. And then following on from this, wanted to see if HIF1 alpha again was involved in this story. And they found that, again, repeating the same conditions from this experiment here, wherein they treated the 231 conditioned media with an inhibitor for lactate, inhibition of lactate secretion, they found that HIF1 alpha was not expressed in TAMs. So using the same conditions forward in the TAMs, they found that ECR activity in TAMs was inhibited, ECR activity in TAMs was dependent on the lactate secretion from the 231 cells. As you can see here, if you use 231 conditioned media that was treated with oxamate, that is again lactate inhibitor, you find that the ECR activity is going down in this case compared to all these other controls. But then this is where the flip side came, was like a little thrown off by the paper from here, because what they did was I haven't included the figure here, but then what they wanted to do was if lactate secretion is actually influencing PU1 binding of HESLA on the HESLA promoter, and that is driving HESLA expression forward. But so what they did was they used luciferase activity to see if the PU1 expression was being driven up, but then lactate didn't actually, they didn't actually see any difference in the rate of luciferase activity or PU1 activity on exposing them to lactate. So they threw out the idea that PU1 was actually driving up the expression of HESLA on lactate stimulation. So they went back to the drawing board and found that there was this other motif in the HESLA promoter for ELK, which is again a transcription factor, which is downstream of the ERK signaling pathway. And they found that when they knocked down ELK, HESLA expression went up. So as you can see from here. So what they justified it by saying that, ELK transcription factor is actually, so lactate is driving ELK to increase the expression of HESLA, but PU1 is responsible for maintaining basal levels of HESLA. They then wanted to see if HESLA is actually involved in stabilization of HIF1 alpha in any way. So if it is actually interacting with any of the other partners or the limiting factors of HIF1 alpha, as I mentioned to you before, like PHD2 and VHL, if HESLA is actually interacting with those to cause an effect. So what they did was they did some IP for HIF1 alpha, and as you can see from here, when the 231 cells were exposed to TAM EVs, but the TAM EVs were themselves where HESLA was knocked down, they saw increased ubiquitination of HESLA. Increased ubiquitination of HIF1 alpha in the 231 cells. And then following on from this, they wanted to see if there was any difference in expression levels of PHD2 and VHL. So in the 231 cells that were exposed to TAM EVs, they saw the expression levels of, they tested the expression level of PHD2 and VHL, again using these different treatments from TAM EVs wherein HESLA was knocked down. There wasn't any difference in expression levels across all of the treatment conditions. But what they followed this up with was doing an IP for PHD2, wherein they found that when the 231 cells were exposed to TAM EVs, wherein HESLA was knocked down, there was definitely some decrease in like the amount of interaction, as you can see from here in the input and in the IP specifically for PHD2, there's more levels of HIF1 alpha wherein HESLA was knocked down. So this indicates that there's definitely some interaction between PHD2 and HIF1 alpha happening. And they followed this up, or they validated this further by doing an RNA pull down assay and an immunoprecipitation assay. So this year is the RNA pull down assay. So when you use probes against HESLA, they pulled it down and they found that PHD2 was actually binding to HESLA. And then this year is just the opposite of, so they immunoprecipitated PHD2 and found HESLA binding to PHD2 compared to the other factors. And they validated this further by using FISH and immunofluorescence. So they did FISH for HESLA, combined it with immunofluorescence for PHD2 in the 231 cells, and they found co-localization. Not sure you can see it. So co-localization of HESLA and PHD2 in the MDM231 cells. Just to finish it off, they did some in vivo studies to see if the same mechanism was holding in vivo models. So they inoculated the 231 cells with or without co-injection of poltams into the mammary fat pads of non-obese or SCID mice. And they found that following on from docetaxel treatment, the tumor volume definitely increased in case of the 231 cells wherein they were exposed to plus the TAM EVs compared to wherein the HESLA was knocked down or HIF1 alpha was knocked down in case of 231 cells. And just following on from there, 6 days after inoculation, they did an immunohistochemistry panel for checking the levels of glycolysis markers for HK2, GLUT1, GLUT3, HESLA, and tunnel. And the same was seen to replicate here in case of the 231 cells that were inoculated with the TAM EVs. Then there was increased glycolytic activity and reduced apoptosis, as you can see here. And then they finished off the paper by using the actual samples that they started off with in the first experimental figure, wherein they found that HESLA levels, so in cases wherein HESLA level was more, they found that to correlate very well with the expression of CD68, which indicated infiltrating macrophages, GLUT1, GLUT3, and HK2. And just to finish off the apoptosis story, they found that HESLA expression was higher in tumor biopsies, which were obtained before neoadjuvant chemotherapy from breast cancer patients, as you can see from here, where chemo-resistant to chemosensitive. And then they found that in chemo-resistant tumors, expression of HESLA was much more. So they commented that HESLA can actually act as a biomarker for chemoresistance. So in summary, after all this, this is a simple model that they presented. So there is some interaction happening between TAMs and the breast cancer cells, wherein high lactate levels actually increase the expression of HESLA. HESLA is packaged into EVs, and EVs are then taken up by the breast cancer cells, wherein it actually stabilizes HIF1 alpha by interacting with the PHD2 and then preventing the interaction of these two. That's it.
Okay, thanks, Sabina, for presenting that. Do we have any questions?
Sorry, that was a lot of data to throw at people.
It's a very comprehensive paper, definitely.
That's my response. It was like, whoa, you were not lying about how many figures. They nutted out every tiny little thing, but I was absolutely relieved at the end where they went back to the tissue samples, because the one thing that, these Nature Cell Biology, pat on the back with that, but the one thing that always worries me about these mechanism papers is they're using one cell line. And that always just creeps me out.
So this was the one thing that I found really, so they were very thorough, and they used multiple experiments to kind of back up what they were showing. And I haven't gone through, like, so in the supplementary data, they have actually done the same experiments using other cell lines as well. So they use BD74, and they've used 468 and MCF7. So like they have done experiments in parallel with other cell lines as well to show the same. So that was the one good thing about the paper. But yeah, it was an interesting paper. I did feel like they kind of were very cherry-picked about looking at long, like lncRNAs. Like, why would you not look at other things in?
That's what everybody does. You do that. You work on long RNAs because you like them. I work on microRNAs because I always have. Everyone picks their favorite RNA type. You got to admit that.
Yeah, like what it kind of like, I think that's like one disadvantage of being like, you're so tunnel visioned into like dealing with just like one thing, and you don't look at like, well, you tend to forget about the other things that can be coming into play. But yeah, I guess that's the limitation of what we do, I guess.
Yeah, I find the same thing. We are always very focused, and sometimes with vesicles, we forget that they are actually a really complicated package. And these experiments where they show one thing and another, and then they assign it to a single molecule or 10 of them of the same. The assigning of function to one thing in a complex package, I always wonder about. But it was a whopping paper.
Yeah, it was like a good, like, like I said, like I was having this, so we had our lab meeting in the morning, and we kind of talking about the same thing. There's so many papers that talk about, oh, we found this, don't know the mechanism yet. I was like, okay, I don't know, what do you do with that? So I feel like that is the biggest problem, like especially with lncRNAs, that is the problem that we deal with like every single day. So like we have these like huge panels that they come up with, like, oh, we screened for this, we found this. I'm like, okay, well, when's the other half of the story coming out? And it never comes out. So yeah, so this was like a good paper, and like they started off from somewhere and actually finished it off at some point. So
the energy to do that, though, I know that like genomic profiling and stuff often seems like stamp collecting, but it's a great way to start. But then sometimes you're right, nobody finishes the story in actually working out the mechanism. And this was a really nice paper. They can't have slept for about 4 years. Like there's a lot of data in there.
Yeah, absolutely. And the amount of like, oh my gosh, the supplementary data is, because the one thing that I kind of did not get, that again, I'm not a protein person, so I don't really know what the take on is about this on other people. They haven't like really quantified their Western blots. So they have like in the supplementary figures, they have like provided all their like biological replicate figures, but I don't know what, yeah.
I think I respect them for not quantifying them, because they can't be quantified.
Yeah, a lot of people would agree that quantifying Western blots aren't really that quantitative anyway, with a lot of error. So, yeah. I mean, when they do quantify it, then some people might even cast doubt on those results. Whereas if you left it, unquantified, then, okay, well, I can visually see and make my own opinions about whether or not it's increased or not.
Interesting. And again, like with this paper, I didn't even know that you can actually deplete EVs using anti-CD81. Do you guys do that?
No, that was interesting to me, because I was wondering, that was one of my questions. Did they try any other antibodies? Because what I was wondering is, Exoquick,
I know, that's why I was like, I don't know how Shri feels about that.
It precipitates everything, and the problem, in fact, the 100,000 G spin as well, precipitates protein aggregates as well. So the fact that they did that CD81, it actually gave me a little bit of security that what they're actually looking at is something that's got this CD81 tetraspan in it, and it's probably got a membrane on it. So that was convincing me.
Right.
but it's a subset. So they still precipitating down a goop.
Yeah.
But by inhibiting that subset, they show the major effect. So that was quite nice. And the Rab27, them following that, proves that it's a membrane brown particle than just protein goop. So I'm forgiving them for using it so quick.
Yeah, so those experiments I found quite nice, that they they use anti-CD81 to actually deplete, and then wherein they actually show the internalization using again Rab27. So that's some very cool experiments. I didn't even know you could do that. It's like, what? This is insane.
Did anyone wonder about the speed of the uptake? It was
45 seconds.
Yeah. That doesn't, I mean, from our work with the bacteria vesicles, we see a peak. We were like, oh, what time point should we do? And we're like, oh, let's go for 24 hours. And it was actually the 1 minute.
Wow.
That was the best one. And they get in very, very quick. So I think people are always surprised at how quick, but yeah, the Rab27 co-localization with the DIO labeled EVs was something nice. I think we should probably think about getting that.
Yeah, because it kind of validates the story quite well as well, that it is not actually because that was when I started reading the paper, I was like, wow. So one question that I had was, obviously when they are co-culturing, right, so they're EVs from 231s and they're EVs from TAMs, and I was like, what? So clearly, yeah.
Yeah, I think any questions I had at the beginning, they did a very good job of answering them as they went through. It was at the end, I was like, it was quite exhausting.
I'm sorry, I apologize.
You did a wonderful job. But it was exhausting.
Because initially I was like, oh, I was trying to juggle really what figures to put in and what figures to kind of skip out on. That was such a tough decision to make, because I felt like every figure told the story. So I couldn't really leave anything out. But yeah, the only thing that I kind of threw me off was the binding of the transcription factor. So they changed from PU1 to ELK transcription factor, but then they didn't really do the experiments that I'd done with PU1 with ELK. That was the only thing that kind of bothered me a little bit. But
I wonder if that was a reviewer comment. Have you thought about my favorite ELK protein?
Maybe.
Last minute experiment.
Yeah. So yeah, but yeah. Any other comments, questions?
So there isn't actually anything more coming through the chat. So if you don't have a microphone or you're not set up, you're very welcome to send us questions through the chat. Or if you're brave, unmute your microphone and ask a question. We won't bite. Just give people a couple of minutes to think about it. Would anyone be interested in presenting a paper next week for our next EV Journal Club, or anyone else interested in chairing one of these sessions? If you could maybe email Anastasia, she would be happy to sort everything. Okay, so Ali is up next week. Great. But yeah, if you would be keen for any other weeks, do let Anastasia know. Other than that, I think if there's no more questions coming through, thank you again, Sabina, and thank you everyone for joining us.
Thank you.
And okay, see you next week.
See you all next week.